introduction. Welcome. So I just going to I want to talk about. So I'm talking a bit about the origin of the RNA world model, and then how we model it mathematically. I'm a mathematician by training, so some of my terminology may be a bit different from what you're used to. I apologise for that. So I want to take a simple model of polymerization or just general cluster growth, and then some techniques for analysing these models. As an example, we'll look at my cell formation, which is what we did a long time ago, and then the sort of real meat of the talk is this stuff on RNA polymerization and modelling that. So I'm going to write down a very complicated model of that, and then look at a number of simplifications. So that's the stage, and then at the end look at an analogy which will help us understand some of the behaviour we see. So this is Leslie Orgel, who did the original work on putting very simple chemicals in a flask, passing electric current through it, heating it up to see what chemicals would form, and found very small quantities of amino acids and other more complicated biological molecules that are necessary for the emergence of life. And so what we're going to look at is the next stage up, so that once you've got amino acids, or that sort of complexity of chemical, how those units then combine to form more complicated structures. So how life works today is that in most of your cells you have DNA, which stores your genetic code. A huge, long, massive amount of information is stored in this chemical data bank. Oh, thanks. Right. But the problem with DNA is that it's so complicated it can't self-replicate. It requires a huge amount of machinery, proteins, to actually replicate itself. And so proteins are these fantastic chemicals which catalyse lots of reactions, accelerate, make reactions possible in a lifetime that you wouldn't otherwise be able to see. So this is really like an executable program. If you think about it in terms of computer science, this is what stores the information, this is what does everything. It produces all the chemicals. And the problem with proteins is that they don't store much information. And so this is a sort of chicken and egg paradox as to which comes first. Do you get proteins which can't store much information, so can't really be a basis for life, or do you get DNA? DNA can't come first because it can't replicate itself. So one possible way out is, as being suggested, is the RNA world, which is a model in which RNA does both of these things. It can store some information and it can also self-replicate. It can be a catalyst itself for chemical reactions. So I'm aware there's a big controversy in the literature over whether this is a sensible model or not, and whether there are other models out there, and probably some of the mechanisms that we write down will apply to other models as well as the RNA world. Okay, so the sort of maths that we need to model polymerization reactions, models I work on are based on Becker-Doring equations, which are a simple model of phase transitions. And the main assumption is that you just have things which grow. So this cluster of CR is a cluster of size R. It's a cluster that contains R particles. It could be a crystal nucleus. It could be a polymer. It could be a collection of stars in a galaxy, or at any sort of scale. And these things can grow by interacting with a monomer and growing with forward rate AR and backward rate BR-1, so assume that this growth process is reversible. And so you get equations like this. So if you think about 4 particles, you can either get them by having a 3 plus a 1 going to a 4, or by a 5 particle fragmenting into a 1 and a 4. And then you can lose clusters of size 4 if a 4 particle separates, or if it merges with another monomer to form the next size up. So you end up with a whole sequence of these equations. I'm not really interested at the moment in space. I'm assuming that all this is happening in some sort of box, and these are all spatially averaged. So you get systems of equations like this. This is a constant mass formulation. We're assuming that there's no input of monomer. You can look at other formulations of Ecuadorian equations where there is stuff put in as well, added to the system or removed from the system. But in general, we tend to look at the constant mass, constant density formulation. So it's quite a nice system of equations that has quite a lot of properties which are intuitive. And, so it's a conserved quantity, the density or total mass is a constant. It has an equilibrium solution. By looking at the ratio of the forward to the backward rates, you can construct a partition function and actually write down what the equilibrium solution will be. And this is in the constant mass formulation. This is a unique equilibrium solution. There is only one. And you can write down a free energy, or the Lyapunov function, which guarantees convergence to the equilibrium solution, provided certain conditions and mapped technical conditions. And this also means that however you start your initial system, because the Lyapunov function must decrease, you're going to tend to this equilibrium solution. And for proving this, there are some identities which we don't need to go into. Okay, but this is a big system of ordinary differential equations, and so to actually get something useful out of it, I'm going to outline an approximation technique, which is quite sort of ad hoc, but leads to some useful equations. So the model we've written down is microscopic. It keeps count of every different size particle. And in general, you couldn't measure a sort of distribution that accurately. So this procedure enables you to get a crude approximation, a sort of macroscopic model of what's going on. So we write down all of these fluxes, these J's from 1 to mass fluxes from one cluster size to the next cluster size, and try and eliminate a lot of these concentrations. So here we've got concentrations of monomer, dimer, trimer, and so on. And we also cross out most of them and just keep, say, every fourth one in this picture. And so we eliminate all of the intermediate ones. And if you do that, after a lot of algebra, you can write down a flux function which depends on this concentration and this concentration, and doesn't involve any of the intermediate concentrations.
So it goes linearly in the chain length, the cost gradient. There's no logarithmic step.
Yeah. Well, no, you can generalize it to make this so that these get wider as you get larger, if you want to say that. But it introduces extra complexity in that you get these extra powers. Because you have to add 4 monomers to go from here to here, this now depends on the monomer concentration to the 4th power. But other than that, the system of equations you get is very similar to the Ecuadorian system we had before. There's these extra lambdas, which in this case is 4, because we've got rid of 3/4 of the information, and ended up with a set of equations that's got most of it taken out, but the same sort of structure. And because it's got the same structure, it has the same properties, that if you think of this as just a system of differential equations, you can still write down the equilibrium solution, and it still depends in the same way on the partition function. You can find a conserved quantity, which is the density or the mass of this coarse-grained contracted system. The system has a Lyapunov function or a free energy, and you can identify these forward and backward rate coefficients with the partition function.
So it's is it exact in
what?
In the final solution, or?
Well, no, it would. No, it's not. It's not exact. I mean, you define the coefficient, so the equilibrium solution is exact. But then you do lose some information because you got rid of it, and you can get the sort of leading order kinetics right.
And also, both the first model and the second model, can that actually be tested in by nucleotide polymerization? I mean, and like for example, the time dependence of the chain growth? Can it be tested?
Well, I'm not aware of any data that would give you. So if you write down the full Beccadorian model, there are so many parameters in it that I don't know of anywhere that you could get details. So if you write down the full, if you worry about clusters up to size 100, say, then you've got 100 forward rate coefficients and 100 backward rate coefficients. And I don't know anywhere that will give you all of those. So in a way, this makes life easier, because if you now throw away 75%, you only have to worry about 25 rate coefficients instead of 100. So it makes it easier to fit models to, because there are less parameters in it. So you can think of it as a sort of renormalization scaling. So we originally wrote this down to try and model these experiments from Louise's group, who was then based at ETH in Zurich, and he was interested in artificial life, in constructing simple cell-like structures, and he had this ethyl caprylate system, which, when you set it up, very little appeared to happen for 30 hours or more, and then suddenly the whole reaction would appear to take place within 10 minutes. And you could measure caprylate concentration at various times, and the concentration of micelles. So she's got a picture. Micelles are these amphiphilic, these molecules which have a head group and a tail group. So a tail group is a chain of hydrocarbons. So this is like oil, it doesn't like being in water, and the head group is polar, so it likes being in water. And if you put these you drop a load of these into water, they will assemble into big spheres with all of the head groups on the outside and all the tails inside. And these things are called micelles. They look like cells. So we're trying to understand this strange kinetics where nothing can happen for a long time and then suddenly the whole lot happens very, very quickly. And so the basic chemistry that's going on is that we have a precursor species, ethyl caprylate, which decays into a monomer at some slow rate, which we'll call K0. And then these things are the things which are going to form micelles. These are the monomers which can aggregate together into larger and larger sizes to form these big structures. And this reaction, this breakup, it happens at a surface, but is accelerated by the presence of the surface. So once we have some micelles around, these CRs, these large clusters, the surface area in the system increases a lot, and so this reaction can happen a lot quicker. So once you've got micelles, you can convert ethyl caprylate into caprylate monomer much quickly, very quickly. But you've got this complicated but fairly quick aggregation of the monomers into micelles. So you can write down a model of this, and it's a sort of slight generalization to the Beccadorian equations, where we have this extra E quantity which is what decays. So this is the decay of the ethyl caprylate, and these same terms appear in the monomer equation as a source term, and then we have the normal Beccadorian system down here for the aggregation of the monomers into the micelles. And we could write down the same structure: equilibrium, density conservation, and free energy.
Why do you have to summarize?
Sorry?
Why do you have to summarize up to the
Oh, well, because I suppose, yeah, okay, so there would be some maximum cluster size that you see as well. It's just mathematically, there's no artificial upper limit on this. in practice, this cluster size distribution would look something like this. So you wouldn't get clusters above a certain size, which is this is typically about 60-ish, 60 to 160 something.
So infinity is fine.
Pardon? Yeah. Well, as a mathematician, you think of 1 as a normal number, and then you think of the 1 as big, and small, small.
And chemically, what is this ethyl caprylate?
It's
And why does it form this C?
It's a precursor species. It's a more complicated chemical which spontaneously breaks down into these amphiphilic molecules.
Uh-huh.
I'm not sure exactly what the chemical formula is for this.
So it's 5 carbon chains. So it's like acid with 5 carbon chains.
Okay.
Uh-huh. Mhm.
So we can go through the same sort of procedure and sort of coarse grain or try and contract this model to write down a simpler system instead of, so if we were to write this down, perhaps we'd have to write down, if this is a distribution centered around 60, we might want to go up to 100 and write down 100 differential equations, and that would be enough. That's a lot. And this coarse grain system has the same properties. But then to actually get a system that we can understand, we can actually do some theoretical analysis on, let's take an extreme case of this coarse grain contraction and just worry about 3 things. So we're just going to worry about the ethyl caprylate, the precursor species, the monomer. So when we do this coarse graining, the monomer has a very special role in the Becker-Joule equations because you only have cluster monomer interactions, so you can't coarse grain and average the monomer with the dimers or the trimers because then you lose the kinetics. So you always keep the monomer separate, so that has its own special equation which we always retain. But the others we can all group together, so everything else, all of this stuff we can group together and just have one concentration of micelles. So with these 3 species, we just get 3 differential equations, which is actually only 2 because we have density conservation. And that system's that simple, you can actually solve, and they very easily, and they exhibit the right sort of behaviour. We have a very slow decay of ethyl caprylate, and down at the bottom you can see the monomer increasing very slowly. The micelles, almost no micelles until very late on in the reaction, and then suddenly the micelles, concentration of the micelles increase, and that causes the concentration of the monomer to increase, and we find this very rapid phase that happens spontaneously. It's all part of the same model after 30 hours of doing very, very little.
That's when n is 60, but for the peak, yeah, right.
And we can test the theory by adding in some monomer. And so you can do some experiments to check the theory by adding monomer, and the effect of adding monomer is to reduce the induction time. So instead of being 33 hours, by adding monomer you can reduce it. And so these are experimental data points, and this solid line is what our theory could predict, and this is somebody else's theory which was around beforehand.
I see. So the other theory was wrong.
Yeah. Okay, so that was an example of using the Becker-Joule equations to model the formation of those very small cell-like structures, which we will return to RNA polymerization. So now, as well as worrying about the size, which we were sort of in mast cell formation. We also need to worry about information content, and so instead of just having CR, we're now going to try and keep track of some information that's stored within the chains, because we've now got different sorts of monomers. If you work with RNA, there are 4 bases, A, C, G and U, and they form these complementary pairs. C always pairs with G and A pairs with U. And so we have a complement, which you get by swapping the C's to G's, or swapping the A's to U's and vice versa. And we're going to make one fundamental assumption that a chain and its complement have the same concentration. So this doesn't simplify the model very much, but we don't lose much of the generality of the model, but it does make it a lot simpler, because rather than having a chain being catalysed by some other chain, the chain is now catalysed by itself, if we assume that the chain and its complement have the same concentration. So we can work with an autocatalytic model rather than having to identify exactly what the complement of the chain is. Okay, and so this work sort of fits in between. So I mentioned earlier Miller, who managed to actually make some of these amino acids by passing electric current through a chamber containing the very simple elements. There's also work by Eigen and Schuster on hypercycles, which is more looks at assuming you've already got a big complicated system of chemicals where each chemical catalyses another element in the network, and then looks at how stable that is and how things can evolve and adapt. What we're doing here is trying to answer the question: how do those hypercycles exist? How do those hypercycles arise? What sort of simpler chemistry can lead to the formation of a hypercycle from a system where nothing existed before? So, the sorts of reactions that we're going to build into our model are these. Okay, so perhaps to go back in a more sensible order, we start off by looking at chain growth, and chains can grow as we did in the simple Becker-Dorian case, where you just have a chain colliding with a monomer, forming the next chain up, uncatalyzed simple polymerization. We're going to assume that happens at some very small rate, and I use an epsilon to denote that, and that's going to always happen very slowly. And then typically you'd expect some sort of hydrolysis. There's always something that works against you, and there'd be some process which breaks up chains and takes a long chain and splits it into 2 short ones. So that is the sort of thing that we're going to constantly fight against. And then, because RNA has the special properties, we're going to build in some of these catalytic things. So one of these is that it has a template-based chain synthesis, where if we have 2 chains here, this one at the length R and one at length S, they can aid this chain of length R growing to length S at R plus 1. So this is a catalytic step where the chains interact with each other and help the growth of one chain. And we're going to split this into 2 sorts. We have the usual, this sort of Watson-Crick accelerated chain growth, where here's our chain of length R, this is this gamma chain, here's our monomer, U, and it's going to bind with some other chain, S, and you see that this is the template, this is the complement of this one, and U is the complement of the next one in this sequence A, so it would be very natural and very easy for that to bind in there. So we're going to assume that this alpha is this autocatalytic rate, where one chain can help its complement grow, but if we assume that a chain and its complement have the same concentrations, this is really autocatalysis. And then we'll also assume that there's this reaction where a different species can form, can help this chain grow. And this is like introduction of errors into the genetic code. So there's a catalytic process. This is going to help chains grow, but it's going to lose us our information because it introduces an error into the code. And then there are even more complicated enzymatic reactions, which are sort of like 3 chain interactions where one of the chains acts as this Watson-Crick template. So this is the middle chain is the one that's growing, this is the template, and a third chain can act as an enzyme to catalyse this growth even more. So these are also going to be fast reactions with some rate zeta.
But that code is not related. I mean, the enzyme.
No.
It's completely
random. This is some sort of, I suppose it's modelling some sort of polymerase enzyme that can just help things grow.
Yeah.
So it just gives some sort of
So that would be normally proteins, so this rather looks like HBAS.
Yes, but the idea of the RNA world is that the RNA
right,
is
of course.
acting as protein as well as the DNA. So I would be quite happy to look at other mechanisms. This is the chemistry as it was explained to me. So overall, we've got a growth rate of this reaction. It can happen on its own spontaneously, it can happen autocatalytically, it can happen because of other chains cross-catalyzing it, or due to this enzymatic process. And I'm sure there are many other reactions going on which we're going to neglect. So I know one is poisoning, where 2 chains can just merge and form a complicated mess, and that takes them out of reaction and can't grow any longer. We're going to assume that the rates of growth are independent of chain composition and independent of chain length, and then write down differential equations for this, which I'm not going to pretend that we can analyse this system of differential equations, but we can actually write it down. So here are some chains, and these are all the rates, and it's just a horrible boring mess. So what we're going to do is look at very simple simplifications, and depending on what question we want to answer, look at a different simplification of that model. So start off with a very simple model problem. Just look at this chain growth with hydrolysis. So that's thing that's going to break up chains, and we've got in these catalytic terms that catalyse growth. And to answer questions like, do we get long chains growing in such a system? There are 2 fundamental quantities that we're going to look at. N is the total number of chains, and N is the total mass of chains, the total amount of material. And then if we take the ratio of M over N, that will give us our average chain length and enable us to answer questions like, do we get long chains or not? So if we ignore the enzymatic process and assume we just have cross-catalysis, not autocatalysis, then our flux becomes this, and we can actually write down a simpler system of differential equations. This is a time derivative of F is the sum of all chains of length R, whatever their composition. We're going to average sum of all different sorts of compositions and just look at this. And this is a system you can solve analytically using generating functions, and you get, instead of having a large system of ODEs, you get a single PDE.
In the regarding cross-catalysis, did you explain that in the previous examples? You explained catalysis and enzymatic reactions.
So cross-catalysis was like autocatalysis, but where the wrong thing acts as a catalyst, and so you get errors.
Oh, there's the Chi thing, yeah?
Yeah.
Yeah. Okay. Yeah, no, it's fine then.
Okay. from a solution, you can actually solve for this generating function, which gives us this mess. It's not a very nice function, but it does enable us to work out explicitly what the total number of chains is, and the standard deviation. And so you get that the average chain length grows exponentially in time, and the standard deviation, the spread of the distribution of chains, also grows exponentially in time with the same sort of growth rate. The standard deviation grows the same as the mean chain length. So you get a very wide distribution of chains, but you do get long chains. But that's sort of what you'd expect if there was no hydrolysis in the model. So that wasn't particularly useful. But now let's put hydrolysis back in. So now we can write down some equations from a model, derive some equations for the total number of chains and the total mass of chains. We've now got our hydrolysis terms back in. These are these eta terms. And we get two sorts of behaviour now, depending on the strength of the hydrolysis term. So because it's just two ODEs, we can solve it. You get the case where if this ratio of hydrolysis to growth rate is less than some critical number, which happens to be about 14, then you get the mass increasing, the number of chains increasing, and the length of the chains increasing, and all going off to infinity as t gets to infinity. But if the hydrolysis is greater than that, then there's some limit. So if we plot the phase planes, so this is a plot of the number of chains across here and the mass of the chains up here. So we start with just monomers, so one monomer, and then you follow a trajectory which goes up and up and up if the hydrolysis is weak. But once the hydrolysis crosses some critical value, there's a steady state here, and you just approach that steady state.
What's the other solution then? The average? No, what's the other solution? Which is also...
Yes. Well, this is a phase plane, so you could start with all sorts of different initial conditions. And so if you started with initial conditions up here, you'd still get drawn to this. If you started with initial conditions up here, you get drawn into this. So these are the trajectories these are the trajectories that you'd follow. So if you started up here, you'd do something like this. So even if you started with lots of long chains, because you've got hydrolysis in the model, you'd still be attracted to that fixed point. So we can get long chains, provided hydrolysis isn't too strong. So that tells us that we can get that these catalytic mechanisms can be strong enough to grow chains, but it doesn't tell us anything about the information content. So we're going to go back and look again and derive a different sort of approximation to look at information content. So we're going to go back and actually do another of these coarse grains things. We're not going to worry about dimers and trimers and lots of different lengths. We're just going to worry about actually two different lengths. We're going to have monomers, short chains and long chains. And short might mean that we could choose it to mean 4 or 10 or 100, and long would be twice that length. So this is just to illustrate the complexity of the problem. We've got 4 different monomers, A, C, G and U. So if we think about chains of length 9, there are actually, if you think about all possible combinations of monomers where the order is important, you've got over 250,000 different chains of length 9. And if you go up to chains of length 10, it's well over 1 million. So, and then if you want chains of length 20, it's 10 to the 12. So these are phenomenally complicated problems to think about the concentration of each different chain type. Even this is not the sort of thing that would be quite tricky to do on a computer. So from our horrendously complicated model, let's just work out what reactions we're going to have in a simplified model. We're going to have normal polymerization, where we just assume in one coarse-grained step, that P monomers can form a short polymer. And that's going to happen at some slow rate. Then once we've got a short polymer, we can add another P monomers, all in one step, and form a long polymer, and that's also going to happen slowly. Our hydrolysis will be that a long chain can split into two short chains, and then our catalytic steps are just repeats of these chain growth, but catalyzed by the short polymers or long polymers. So we have the same sorts of terms in our kinetic equations. So we have the same sort of equations crop up again. So we want to know, does this system allow us to get longer chains? And then do we just get a few chains, a few different types of chains, or do all chains grow? So the simplest solution to look for is one in which all the different types of chains grow with the same concentration. We've got the same monomers. We're going to assume all our different monomers have the same concentration, and all our short chains have some other concentration, and all the long chains have another concentration Z. So now, instead of having 250,000 different differential equations for our Y variables and 10 to the 11 chain concentrations for our long chains, we just have 3 ODEs, one for the short chains, one for the long chains, and one for the monomers. And this system we can analyse using asymptotics, because we've got a small parameter. The spontaneous polymerization is much slower, so it's much slower than the autocatalytic polymerization. And so at early times we can generate this solution. So this epsilon is a small parameter, and in comparison to these alpha and chi. So the sorts of solution we get looks like this. So this is telling us that as here epsilon over alpha, epsilon is much smaller than alpha, so this is a small concentration. So in general we have a low concentration of polymers, but if we look at how this evolves in time, it's 1 minus some constant times the power. So this is something which is going to grow. These are is something which starts small, but looks like it's going to have a blow up at some critical time. So we can use the form of this solution to estimate this critical time. So it looks a bit like these micelles, where everything stays very, very small for a long time, but then suddenly, after a long time, something starts to happen, and it happens very quickly when it does. So if you wait long enough, you will get a larger concentration. And we can work out roughly how long this is. This was an assumption, though. This was based on assuming that all different types of chain have the same concentrations, and that may not be the case. So to see if that's a sort of solution that you would actually see in practice, we should also look at the stability of this solution, and so we do that by linearizing around the solution. So usually you'd linearize around an equilibrium or a steady state solution, and to look at the stability and see whether perturbations grow or die. But formally you could do the same sort of thing with a kinetic solution. So this is the solution that we've written down. This is this sort of solution where very little appears to happen for a long time, then suddenly it blows up. This is our solution for chains. We're now going to reintroduce our m's and n's. This is not the length of the polymer, but this is the information in it. So this enumerates the 250,000 different short polymers. So we're going to assume each short polymer has this concentration, but is perturbed by this amount yn hat of T, and similarly for the long polymers. And then look at the stability to see if this solution is stable or unstable. So you can plug this into the equations. I won't go into all the gory details, but what you end up with is equations for how these perturbations evolve in time. So we know what this uniform solution, how this behaves in time, and that feeds, this information about the uniform solution feeds into these coefficients A, B, C and D. And now we want to do a sort of piggyback problem and look at how these hatted variables evolve through time. And you can make some simplifications that if the average of these perturbations is 0 at some point, then it's always 0 afterwards. And the other thing we need to do is look at only particular sorts of perturbation. So we're going to seek a, this is a, I should put quotes around this, it's not. Really, this is some complicated function that involves the information and time. So different information can grow at different rates. Polymers containing different types of chains can grow at different rates. And we're going to assume that we can split this up into one part that contains the information and another part that contains the temporal evolution of that. And we need a further approximation, a further assumption that this perturbation for long polymers is just the sum of the ones for the two shorter polymers. And then we can look at how these perturbations evolve in time, and whether S and L grow or decay will tell us whether the uniform solution is stable or unstable. So let's get down to these two equations for the short. This is the time evolution of the perturbations of the short ones, and this time evolution of the perturbations of the long ones. And we can determine whether this system is stable or unstable just by looking at the sum A plus C and the product AC and the comparison of 2 BD. So this is the trace and the determinant of the matrix. And the Ruth Hilbert's criteria tell us these stability conditions, and you can analyse the different cases. There are different cases where, depending on whether you have constant mass or constant density Bechdelian equations. So an instability from these that you can get down to one condition is this, that the alpha S has to be greater than n times this. So this is quite intuitive. Well, this tells you this is the autocatalytic process. So it says that you're going to get an instability if the autocatalytic process is greater than a combination of that, which is a cross-catalytic process, and epsilon S, which is the spontaneous polymerization rate. So this is what we say we assumed was small. And so it says that basically if the autocatalytic process is strong enough, then you would get an instability develop.
So the autocatalysis really corresponds to this template.
Templating, yeah. Which is intuitively what you might expect, but it's nice to actually see it come out of the equations, out of the model. So you can look at various special cases, because the model is even now so complicated, and get similar conditions on the autocatalytic process of the long chains as well. And there's another more complicated instability, which is due to hydrolysis. So initially you think hydrolysis is a thing which you're fighting against because it breaks up the long chains into short chains. But actually a small amount of hydrolysis is good for your system, because catalysis requires the presence of other chains in order to promote growth. And what hydrolysis does is increase the number of chains, because it breaks the long chain into two short ones. So having a small amount of hydrolysis in the system is actually accelerates the growth, but obviously not too large. I guess that's all been. So really, trying to think about what this actually looks like. So it's a sort of symmetry breaking, but instead of thinking about it in space or in left and right, you've got this information space which has a really high dimension. So I was trying to think of a way of illustrating this using a more intuitive process. And a thing which I think it's similar to is solving a heat equation, or thinking about heat generation. So this is a sort of toy system for what might go on inside a furnace reactor, where you're generally, the simplest mechanism is you've just got this linear growth, d theta dt is theta. Things are just going to, temperature's going to increase exponentially. But then autocatalysis really says that if you've got some temperature around, if you've got some heat around, then the temperature's going to go up much more rapidly. So instead of just growing with theta, it's going to grow like theta squared. That's what we see in the chemical reactions. Instead of having just C dot is proportional to C, you have C dot proportional to C squared. But then we also have this cross-catalysis, which is really where other things catalyse the growth, and that really spreads out the information. So that's a bit like a diffusion operator. It spreads things around. It doesn't decay, destroy it, just moves it to other chains, other types of chain. So, in a way, this system has similar features. And this is something we can actually draw pictures of, so we can try and get some intuitive idea of what's going on. So this system, space is like our information space. So we can have a uniform solution, and this does mean spatially uniform, the same temperature everywhere in space. And such a solution will still blow up because we've got autocatalysis. So, and you can work out the time at which it blows up. So this sort of solution will look like this. It's just the same as this picture here. So this is space down here, x, and time across here. So you start with some small temperature, it grows very slowly, linearly, and then it's going to blow up.
So blow up corresponds to a peak in your theta of x space, or what?
Yeah. Right. Yeah. So yeah. So it's actually a divergence in this. In our other model, it would just mean the concentrations have got very large, and you'd have to do a rescaling and look at the next time asymptotically. But once, I mean, that's the main question we're trying to answer is, can you get large chains? Unless it's blown up, then the answer is yes, you can get really large chains, large concentrations of one chains. And then you want to know, is that solution stable? So you think about fluctuations. So you introduce, so you think about, so I should really put this as an initial condition, theta of x naught, and think about what happens at theta at t equals naught, and put in some fluctuation. So you have the constant temperature, which is this 1 times this, but plus a small perturbation that would look like sine kx. And then you'd ask, does that evolve? How does that evolve forward in time? And it grows. And different perturbations have different growth rates. And so, if you have a small perturbation, it would grow like this. So here, this is a almost uniform solution, but it's got a small perturbation. You might be able to see wiggles here, but as it grows forward in time, you see the wiggles get larger oscillations. And you perhaps can see 3 oscillations here, but actually the middle oscillation is slightly larger than the 2 side ones, which is obvious at the end, but is almost impossible to see at the beginning. So those oscillations are accentuated by the presence of autocatalysis. So what we can do is compare these two. Okay, so that's our solution with the oscillations, and this is our solution with a genuinely uniform initial condition. You see, both solutions are going to blow up eventually, but this uniform solution would blow up a bit later than this one that has the oscillations in it. And the same sort of thing would happen in our I would expect to happen in our RNA model, that you wouldn't see a uniform distribution of all types of chain, but there would be some random fluctuations due to the environment that would cause some chains to form and not others, and those chains would then proliferate.
I mean, that's probably kind of typical. In many cases in physics we have dispersion relations where the growth rate again is small for k equals 0.
Yeah.
And so then there will be a peak at some value.
Yeah. But it's interesting to know it would be interesting to know what causes that peak, whether that's a random fluctuation due to the environment or whether there's some deterministic process which causes the peak. So, just to summarise, we've written down a very complicated model for RNA polymerisation. It involves lots of different processes, and the problem is computationally immensely difficult using all the different types of chain that could form. And non-linearity prevents you analysing anything analytically. The combinatorial size prevents numerical solutions, so it's a really phenomenally difficult problem. But by using some ad hoc approximations and some analogies, we've hopefully illustrated that it does have the right sort of properties, that it allows long chains to form, that you can conserve and propagate information forward, and that you need this autocatalysis in order to get these chains to proliferate and to remain. And there are some surprising results, like hydrolysis not being always bad, but actually helping you if it's weak enough. Okay?
Thank you. Questions, please?
Yes, I understand that you have to make these certifications because of these reasons that you mentioned here. But how you validate these certifications? I mean, just the fact that you can reproduce a curve going up like that, is that really sufficient for validation of your certifications?
Yeah, I get it. Well, I take your point. Yes, and if it was, so one of the problems we haven't really tackled at all is parameter estimation. Is trying to work out what values to assign to all of these parameters. And so if there was experimental data that enables us to get parameter values and to fit these sort of curves, then that would be very useful, but it's very difficult. If you know the data, I think what we could test it against, it would be very interesting to know.
For example, the template-directed polymerization of RNA, of course the DNA, there are of course lots of experiments by Joyce in the 80s maybe. Can we use those experiments to verify model assumptions?
I could, so I've not studied that.
One of the curious things one found back then was, I mean, not curious I think, but typical things is typical chain length was never more than 20. Then they stop a little bit.
Yeah, I mean that's the sort of thing that I would imagine that it sort of gets at 10 or 20, and then that's probably long enough to form some sort of hypercycle, and then Huygens' model is perhaps more relevant than this would be.
In fact, you said it was Orwell in the first picture, but it wasn't maybe Stanley Miller instead.
Sorry, no, I got Miller written down.
Yeah. Okay. So you have this assumption that the sum of what was that? The information contained in long polymers could be split up into the information in two separate polymers.
Yeah.
That's an amazing.
Well,
it works, you think, yeah?
That was a technical So there's sort of technical. So we want to know how these perturbations evolve in time. And so you've got 250,000 of these, so you can't solve that problem. So what you do is say, right, let's only consider certain perturbations. We want to consider the stability of this. So we could consider all possible perturbations, or we could consider just a small subset of perturbations. And that's really what we're doing. It's just considering a small subset of perturbations which happen to obey certain properties. So that's still quite a large. But yeah, so what we've done is really say we've got some sort of parameter space. So in general, you've got some sort of large dimensional parameter space, and we've said, well, if you're in this region, you're definitely unstable. Because the perturbations we've considered tell you you're unstable. Now, the perturbations we've considered might be stable this side of the boundary, but there might be other perturbations which are unstable on that side of the boundary. So what we've done is construct a region where we know there are unstable solutions, and uniform solutions unstable. But actually, it's probably bigger than what we estimated. So actually, these the inequalities that we write down are actually very strict, and actually even if you break those inequalities, you still might be unstable.
Yes. Please.
You consider two pictures, yeah? With some perturbations and without perturbation, two cases.
Yeah.
And then can you see some, let's say, interaction between these perturbations? So can they influence on each other? Because just it wasn't clear from the graph, they were like some parallel tube, some tubes, or is it included in the model, or these several perturbations which you can include for your consideration in your case, and they in some way interact with each other or influence on each other?
Yeah. Well, yes, at later time. So this is a linear problem.
Yeah.
So in this case, the perturbations wouldn't interact, but
because they're not interact in this case.
Yeah. The perturbations wouldn't interact, but the perturbations would have a growth rate, and so the perturbation you would see in an experiment is the one that would have the largest growth rate. So if we did try and find what these perturbations, so each of these different sine NNs would have a different growth rate. And so you'd look for the one which had the fastest growth rate, and that would be the one that you should see in an experiment.
So the simplification that you used earlier, that would not, I guess, obey your linear analysis anymore. That would violate this, would it not?
Which thing?
That the sum of the information of two smaller chains is related to the sum of the information of the large chain.
Well, that's... So we consider a family of perturbations, and in that family there'll be one that has the fastest growth rate. There might be another perturbation that's outside that family that we consider that has an even faster growth rate, and it might be that one that you've seen in an experiment. But there'd probably be one in our class, in our family, that was quite close to one that might look similar.
Yes. Right. Any further questions? If that's not the case, then let's thank Jonathan again. And let me introduce one more arrival today. Is Sarah Walker, just arrived today. And she was hit by a car accident just before coming, not related to your arrival, I guess.
No.
Okay, so we... Oh, that's one.